Hello and welcome to another ACHEMA Highlight Webcast, where we take the chance to give you some of the points, introduce you to some of the topics that shape and change the process industry, and that will, of course, be covered in much more depth at ACHEMA 2024, which will take place from June 10th to 14th right here in Frankfurt. So, who would have thought a couple of years ago that we would be talking at the coffee table about the methods for vaccination and the workings of mRNA? Well, we did over the last couple of years. And this is only the tip of the iceberg when it comes to pharmaceuticals. There's a big change in pharmaceuticals taking place. I would be about to say a revolution, because we are encountering new kinds of drugs, completely new mechanisms, completely new workings, especially in the field of gene and cell therapeutics. So what's so different about them? Well, some things that really spring into the eye are they are much more individual and personalized, they're very complex, and they are very, very expensive. So what can we do to better exploit the potential of these new kinds of drugs? It's my pleasure to have an expert here today whom I can ask exactly these questions. Welcome to Sadik Kassim. He's the Chief Technology Officer in Genomic Medicines at Danaher, an immunologist by training, and he has been involved during his whole career in the development of cell and gene therapies, and he has been an active player in the development of 3 out of the 6 currently on the market available CAR cell therapies. So welcome, Sadik, to our webcast today.
Thank you for having me. Thank you for the invitation. It's my pleasure.
Sadik, a lot of our viewers are probably chemists or engineers who are familiar with the traditional way of producing drugs. So producing an active ingredient, formulating something, making a pill, giving it to the patient. What is the basic difference with these new kinds of drugs we're talking about today?
Yeah, it's an important question. I mean, I think there are 3 main differences between traditional small molecules and the cell and gene therapies that are in the pipeline today. The first major difference is that we don't really have industrial equipment or capabilities for manufacturing these therapies. A lot of these therapies came out of academic institutions, and so the manufacturing processes were very much designed for academic trials to treat 1 to 2 to 3 patients at a time. And now that we have commercial approval of several of these therapies, we have to repurpose or come up with new approaches, new manufacturing process that enable the industrialization and the scaling of these therapies. So I think one major difference between cell and gene therapies and traditional small molecules is we don't have the manufacturing processes that enable industrialization and scaling today. The second is an incomplete understanding of the analytical methods for characterizing the active pharmaceutical ingredient. So many of these cell and gene therapies have multiple mechanisms of action. And so something as simple as a potency test ends up delaying or limiting our ability to commercialize these things, because we don't know necessarily what the active mechanism of action is, how these drugs work. And so oftentimes what you end up having to develop is a matrix type of approach towards characterizing and measuring different potency attributes to really release the product. So I think from an analytical perspective, there's also an incomplete understanding of how these therapeutics work. And then I think the third major difference relates to many of these therapies are being developed at a different sort of scale for patients. So rather than one manufacturing batch being able to treat hundreds to thousands to millions of patients like we do with small molecules, here one manufacturing batch oftentimes treats 1 patient. So it's a very custom manufacturing process, and that customization brings with it its own set of challenges. So I think those are the 3 big differences between small molecules and cell and gene therapies.
Mm. And I guess all three factors also contribute to the fact that those drugs are today so very, very expensive, which is really a problem, especially if we think about treating, well, common forms of cancer, for example, where we have a lot of patients and need a lot of drug. What are the main factors we could target to decrease the cost of these drugs?
Yeah, and cost is a big factor that has limited the real commercial traction of these therapies. And I think the three main levers that are impacting cost are number one, labor costs. So because these things are highly custom, and because the manufacturing processes have come out of academic institutions, many of the unit operations are manual. So you need a large labor force to actually be able to produce these drugs. So people are moving towards automation of the unit operations, but still, I think labor costs tend to be, one of the major costs here with producing and manufacturing these drugs. The second major cost is really capacity utilization. So with a traditional small molecule, or even with a biologic drug like a monoclonal antibody, you can schedule the manufacturing capacity based on demand that's often projected years in advance. Here, because you're manufacturing a product oftentimes for a single patient, utilizing the capacity that you have becomes a really tricky dance. So you can maybe overbuild manufacturing capacity, but underutilize it, which will lead to higher costs. Or maybe you have less manufacturing capacity, so you're unable to address the market need. So I think manufacturing capacity is the second major driver of cost. And then the third one is the cost of the critical raw materials. So the ancillary materials to manufacture these therapeutic products. Because oftentimes the critical raw material or the ancillary materials are just as complicated as the drug itself. So, for example, when you're making a CAR T therapy, oftentimes one of the critical raw materials is a viral vector. So first you have to make a virus, which is a really complicated raw material, and then you use that virus as an input to manufacturing the final drug product. So the cost and the industrialization of the critical raw materials is a third factor that's driving costs. And people are working towards industrializing the raw materials to reduce the costs. But I think, there's still a ways to go before we, can make it really cost effective. But I think those are the three major levers that are driving the cost for these therapies.
Mm. And I think especially the aspect of individualization probably is not only a thing about cost, but also something you have to think about rethinking the value chain, actually, because nowadays, if somebody produces a drug, they sell it, it goes well to the pharmacist, you and I get the same drug. It's a kind of one-directional. In this case, there has to be some feedback loops with the patient. So how do we have to rethink the value chain? Who has to do what or cooperate with whom?
Yeah, it's a really critical question. And I think, oftentimes, like you said, you get a drug from a pharmacy, and it's the same drug that's produced in Germany, that's produced in the US. There's very clear defined release specifications. And it's the same with cell and gene therapies. But the difference is that a lot of these therapies are administered in the hospital setting. So what we really require is a closer integration with healthcare facilities like hospitals and like the prescribing physicians, to really close that loop. The second aspect here is, rethinking the value of these therapies. Because oftentimes, the excitement with cell and gene therapies is oftentimes these are potentially curative therapies. So in the case of, for example, hemophilia, we have a couple of AAV gene therapies that are on the market today, and they completely replace the need for factor 8 or factor 9, infusion. So rather than getting a monthly infusion of a drug, a one-time shot could potentially cure the patient. So, the one-time cost of these therapies tends to be really expensive. But over time, if you look at the cumulative life span of the patient, because you're potentially curing them, it actually ends up being much less expensive. So I think another, player that needs to be looped into the value chain are the actual payers themselves. The healthcare systems have been designed to support therapeutics such as small molecules and biologics, which require repeat constant infusion during the lifetime of a patient. So the cumulative costs tend to be really high, but the one-time cost, on a regular basis is lower. Here, the complete value proposition of cell and gene therapies gets completely flipped over. What you're saying is that a one-time upfront cost, fairly high, will reap long-term benefits in terms of value to the system. But the system, from a payer perspective, isn't designed to really support these drugs. So I think in addition to the healthcare providers, the systems, we also require the integration of the payers themselves for them to reevaluate and maybe reformulate how they value these therapies relative to traditional small molecule and biologic drugs.
So a lot to think about, a lot of exciting perspectives, I think, and a lot to discuss at ACHEMA as well. So we will continue the discussions there, especially in the highlight session. And thanks for taking the time to be with us today. And we'll see you again at ACHEMA.
My pleasure. Thank you for having me, and I look forward to seeing you and everyone at the meeting. Thank you.
And if you want to hear more from Sadik Kassim and from other experts on these exciting perspectives, then you should mark your calendar now for Monday, the 10th of June, where we have the pharma highlight session at ACHEMA. Of course, pharma will be a topic throughout the whole week on the stage, in the sessions. And if you are thinking about starting a new pharmaceutical production, you will find everything you need, all the equipment for developing drugs and producing them in the exhibition. So make sure to schedule your visit right now, and we will see you at ACHEMA 2024.